We're starting discussing protein synthesis. This is how our bodies take our DNA code and make it into our phenotypes. And it's a two-step process called transcription and translation. It's going to take us a little while to get through this. One of the harder ideas in biology to understand. But let's just dive into it and see how we do. So first of all, what is transcription? Transcription is a process in which part of the nucleotide sequence of DNA is copied into a complementary sequence on an RNA strand. And that RNA is called messenger RNA. So we don't go directly from DNA right to proteins, right to phenotype. We have a middleman. The middleman is RNA. And we're going to take a look at some differences between DNA and RNA as we go into this. Transcription is necessary to take the DNA code found on a gene outside of the nucleus. We have a location problem. The DNA code is in the nucleus, but proteins are made by ribosomes out in the cytoplasm. Then they go to the rough endoplasmic reticulum and to the Golgi apparatus. RNA is necessary. It gets made in the nucleus and brings that DNA code out into the cytoplasm where the proteins actually get made. So we'll get more into RNA and we'll get a better understanding of it as we go. And I know we already talked about this, but RNA is very similar to DNA, and there's three main exceptions. Number one, RNA is single-stranded. That makes it a lot more fragile, so its lifespan is not nearly as long as DNA. We find DNA evidence in fossils and crime scenes and things like that. We never find RNA evidence because RNA breaks down a lot easier because it's only a single strand. There's no thymines in RNA. There's uracil instead, and it has the sugar ribose, where in a DNA nucleotide the deoxyribose would be found. The only difference between ribose and deoxyribose is that deoxyribose is missing an oxygen, where ribose has that oxygen. So that's where the name deoxyribose comes from. I'm just going to give you a lot of information right now, and we'll work with it as we go, but there are three types of RNA, and they're named for what they do. Messenger RNA carries the code found in a gene outside of the nucleus, so that information can be used in the cytoplasm to make a protein. It carries the DNA message from the DNA in the nucleus out to the cytoplasm where the ribosomes are. That's why it has the name messenger RNA. Transfer RNA, we're going to get to when we do part 2 of protein synthesis, and that's called translation. Translation is when you translate from the language of nucleotides into the language of amino acids. So transfer RNAs are used in the process of translation, and they carry the, amino acids. They carry the amino acids to the messenger RNA, so they can be put together to make the proper protein. So they're transferring the building blocks over to the ribosome. And finally, we get ribosomal RNA. Ribosomes are not one of those membrane-bound organelles, so that's why prokaryotes have ribosomes as well. Ribosomes are made of ribosomal RNA and protein. So that's why prokaryotes are able to have them, because they're not a membrane-bound organelle like a lysosome is or like a Golgi apparatus is. Let's dive right into transcription here. The word transcription really means to take something and rewrite it. And that's what we're doing. We're transcribing the DNA into another kind of nucleic acid called RNA. Remember, the whole purpose of this is because DNA is stuck in the nucleus. We don't make proteins in the nucleus, though. So the message on the gene has to be copied and taken outside of the nucleus to the cytoplasm where the ribosomes actually get made. Where the proteins actually get made by the ribosomes. Step 1, we have RNA polymerase. That's an enzyme. It ends in ASE. It's going to bind to an area of the gene, of the DNA, called the promoter. So RNA polymerase sounds a lot like DNA polymerase. DNA polymerase goes in that process that we call DNA replication, that takes DNA and copies it. RNA polymerase is a polymer that makes RNA. So it has a job. It's a lot like DNA polymerase, but we're making RNA with it instead of making DNA. And we're going to get into what those differences are as we go. This picture down here shows you a good example of this. Here's your DNA. We have the promoter right there. That's like the starting line. The RNA polymerase needs to know where to start copying. We have 20,000 some genes. So the promoter lets the RNA polymerase know where the beginning of the gene is, where it has to start doing this process called transcription. All a promoter is a region of DNA. It's a section of adenines, thymines, guanines, and cytosines. They're put together in a certain way, maybe a few hundred nucleotides long, and it signals the RNA polymerase that this is where the gene starts. Alright, step 2. DNA polymerase is going to unwind the 2 DNA strands just like DNA polymerase did by breaking those hydrogen bonds between the nitrogen bases. And finally, as we keep going here, RNA polymerase is going to follow the base pairing rules of DNA as it adds complementary RNA nucleotides to one of the DNA strands. You have to remember, RNA is single-stranded and does not contain thymine. It pairs with another nucleotide called uracil whenever it comes across an adenine in the DNA. We'll practice this quite a bit in class too. We're just going to get the basics down right now. Alright, as the RNA polymerase goes down the strand, it's only using one of the DNA strands to make a single-stranded RNA. Remember, in DNA polymerase with DNA replication, the DNA polymerase, you had 2 polymerases using both strands. We don't do that here. One strand's called the coding strand, so that RNA polymerase goes from the 5' to 3' direction using that strand to make a single-stranded RNA molecule, only one of them. As it reads that DNA, the DNA closes up behind it, and the promoter told the RNA polymerase where to start. Well, there has to be a sequence that tells the RNA polymerase where to end, and that's called the terminator sequence. The terminator sequence is just another series of nucleotides that tells RNA polymerase the gene is over, stop transcription, you have a copy of all the information. Here's a picture of what we're talking about. Up top here, we have a double-stranded DNA. And down here, we're showing you the bottom strand, kind of like in this brownish tan color. And you know that's DNA. It's one of the DNA strands because it has A's and T's and G's and C's in it. So this strand is the only one being read by the RNA polymerase, and it's being used as a pattern to make the messenger RNA. So here's an A. A's go with T's in the DNA, but we don't have T's in RNA. So this A goes with a U. RNA polymerase then reads the T. T's can make hydrogen bonds with A's. It reads a G. G goes with C's. Reads a C. C has to go with G. Reads a T in the DNA. Makes an A in the RNA. Reads an A in the DNA. That makes a U in the RNA because there's no T's in RNA. And it starts from the promoter way out here, keeps following the base pairing rule till it hits the terminator sequence. And that's how an RNA molecule is made from a DNA gene. Just a little reminder here, a gene is a sequence of DNA that can make an RNA or a protein molecule. Genes are functional. Not all of our DNA is genes. A gene, and even DNA in between genes, there's really 2 parts that it breaks down into. There's exons and introns. Exons are DNA sequences that actually make the code that goes into the RNA that gets used to make the proteins. The proteins could be anything from A-type blood to the melanin in our skin to cell parts, those sorts of things. But scientists also discovered there's these other parts of our DNA called introns, and they're just made of A's, T's, G's, and C's just like the exons are, but their sequence is in such a way that it's not useful. It's almost like gibberish in DNA. They don't provide a useful code to make a protein. There's a lot of theories as to why introns are even there. Why are we carrying around DNA that isn't really coding? It doesn't help us make proteins. There's got to be use for it. I just don't think we've discovered it yet. And there's a lot of theories out there as to why introns exist. But I want to tell you about what has to happen with those introns. We just learned about transcription. Start at the promoter, you follow the base pairing rules to make an RNA molecule until you reach the terminator. But when transcription happens, the exons, which is useful information, and the introns, which is that junk, unknown, we don't quite know why it's there DNA, they're both represented in that messenger RNA molecule. We have to do some editing. So after transcription, the mRNA gets edited. The parts of the mRNA that should say transcribed over here, that were transcribed from intron sequences get cut out from the mRNA and not used. It'd be kind of like if someone's making a movie, they have a whole lot of film, an editor has to come in and they cut out certain scenes because they're not necessary to tell the story of the movie. That's what introns are like. They're DNA sequences, they got copied into RNA sequences, but they're not helpful. They're not supposed to be there. So they have to get removed so that the mature RNA is all spliced together and all the junk has been cut out of it. That's what I have down here for you. Again, I should say transcribed. Here's a picture to show you. Up here, we have our DNA, and the darker colors are exons and the lighter colors are introns. So here we have a DNA molecule as 1, 2, 3 exons with 2 intervening introns, which again aren't providing any usable information. So what happens, we do transcription and we make RNA, which is this green molecule. We made RNA out of the exons and out of the introns. But we can't make a protein out of this. This is called the primary transcript. It's like when you write a paper for English class, you have a first draft. It has a bunch of stuff in it you're probably going to take out, and you're probably going to rewrite. That's what these little light sections are. These are the parts that you're eventually going to take out because they're not providing anything useful for your paper, but this is just your first draft. Down here in step 2, you're going to do some editing, just like our bodies do. Our bodies come and they cut these introns out, and as they cut them out, they splice these 1, 2, 3 introns together down here into this molecule. This can be called the mature messenger RNA because it has only the information needed to make that protein. And then this leaves, that's what this arrow is showing you, this leaves through a nuclear pore and goes out into the cytoplasm. Out in the cytoplasm is where ribosomes combine to it, make that immature protein, that polypeptide that can go to the rough ER and the Golgi apparatus and then go do its job. Well, right here, this is the info on how to make that protein. After the mRNA cutting and gluing, we call that mRNA splicing, the now mature mRNA leaves the nucleus and moves through a nuclear pore and goes out into the cytoplasm. Out there is where step 2 of protein synthesis occurs. That's called translation. Translation is using the code found in the mRNA as well as a ribosome and transfer RNAs to make the protein. Actually, it's the immature version of the protein because it's supposed to progress through some organelles to become mature. Alright, we'll start on this. We have a couple minutes here. Translation is the second step of protein synthesis. Some basics. Here's a definition for you. It's the decoding of an mRNA message into a protein chain. Maybe even we should say an amino acid chain there. Again, we just said this. There's 20 different naturally occurring amino acids. Those 20 amino acids put together in different amounts and combinations make all the proteins that make the phenotypes of organisms. Polypeptides are those immature protein molecules that have to go through the rough ER and the Golgi apparatus, and that's what we have here. So this is kind of like an overview of where we're going to be going with this. We're going to decode that mature messenger RNA into an immature protein chain that is made of amino acids. And then it will then go through the ER and the Golgi to become mature and do its job in the cell or in the organism's body. This is what we need to do translation. We need an mRNA that contains the message from the DNA. That's the instructions. We need many transfer RNAs. Each one will have an amino acid on them, and they'll bring over the amino acids when they're needed so they can be put together peptide bonds to make that immature protein. And then we need the ribosome to facilitate everything. The ribosome is going to read the mRNA and then bring in the right tRNAs to put the amino acids together the right way. And that's probably a good stopping point for us. We'll get more into translation as we go.